I will be your host today, and thank you very much, Rafael, for being here virtually in Austria and in the entire world. This talk will be streamed in the worldwide Neuro. So for all of those who would like to ask questions, I'll be looking at the chat box. You can post your questions and then I ask them at the end. Or we can also at the end have a session of Q&A, and I can bring you in and you can ask the questions personally if they're not too many. This said, I'm going to go offline right now, at least not turn off my video, and Michele will present Rafa. Thanks again, Rafa, for being here.
My pleasure.
Okay. So thank you, Max. And I'm very happy to introduce Professor Rafael Yuste, who was invited on behalf of students and postdocs at IST Austria. Rafael Yuste is Professor of Biological Sciences and Neuroscience at Columbia University. He has started by studying medicine in Madrid, and afterwards he has been for a while in Cambridge in Sidney Brenner's lab. He then went to Rockefeller to do a PhD with Larry Katz in Thorsten Wieser's lab, and afterwards he was a postdoc in David Tang's group at Bell Labs. He joined Columbia in '96 as assistant professor, got tenured and full professorship within a decade, and he's been there ever since. He's also co-director of the Kavli Institute for Brain Circuits and director of the Neurotechnology Center at Columbia. He's very accomplished and has received many awards throughout his career, in which he has pioneered the development of calcium imaging of neural circuits, two-photon imaging and optogenetics, and more recently, holographic microscopy. The main focus of his research is on neural circuits and the functioning of neurons as an ensemble, which is, well, also the topic of today's talk. And with this, I want to thank again Rafael for accepting the invitation, and I think we are all looking forward to hearing your talk. Thank you, the stage is yours.
Thank you, Michael, and thank you all the students and postdocs and the faculty of the IST for the opportunity to speak to you. Greetings from this epicenter of the epidemic from New York City. In fact, I'm talking to you from this building right here. I don't know if you can see my cursor. Actually, this is sort of, this is the room where I'm speaking from right here. So those of you who care for maps, this is the view from Columbia University looking south. This is Broadway, the Hudson River. So this is Midtown Manhattan. And I have good news from the point of view of public health. Situation in New York in the epicenter is, it's not too bad. Things are under control. In fact, there's been a little bit of exaggeration in the media, in which they reported in individual cases as if everything is a disaster, and things have not been that bad. Although the economic crisis is probably going to be a disaster, at least from what I can see. So medically we're fine, economically we'll see about that. No? Anyway, my talk today starts with magnets, okay? And it turns out, you probably know, if you take a magnet apart into the individual atoms, the atoms are not magnetic. But when you put them together, something happens, some magic happens, and the system becomes magnetic. And this, 100 years ago, this gave huge headaches to physicists, because how can you get something out of nothing? If there is no magnetism in the individual atoms, how can magnetism arises or emerges from a collection of atoms? And this is a classical example of what we call in science an emergent property. An emergent property is a property of a system that has many parts, which by definition is not present in the individual components. And it's not black magic. The emergent properties arises from the interaction between components. And obviously, in order to interact, you need to be with other components, otherwise there's zero interaction. So magnetism was actually cracked, or ferromagnetism to be more specific, was cracked by this young German Jewish grad student, Ernst Ising, who in 1925 proposed a model, the Ising model, He was actually given the problem of understanding magnetism by his PhD advisor, and he couldn't. He didn't understand that it was supposed to be hard. So he said, well, this is a very simple, straightforward problem. No? If the atoms are interacting, he drew this equation. He came up with this equation in which the S is the spin of each atom. So Si and Sj are two atoms that are interacting. The J is what he called the coupling coefficients. In other words, the strength of the interaction between the atoms. And if the spins are coherent, if they're both positive or both negative, and there's a significant J, this quantity, this multiplication gives you a sizable number. And he then said, okay, if we sum across all the interaction between all the atoms, we can define a quantity called energy. And he made that quantity negative. Energy represents in physics the propensity that a system has to change. So if you have a lot of energy, you're likely to change a lot. Now, if you have very little energy, you are very stable. So he said, the more interactions, the bigger this number, the more negative the energy, which means the more stable the system is. And with this equation, he predicted the existence of groups of atoms that were called spin glasses. This is an example of some measured spin glasses in two different territories with two different spins. And these spin glasses would be stable, and because they would be the system of the lowest energy. So this was a breakthrough because even though Einstein never got the Nobel Prize, in fact, he was chased out of Germany by the Nazis. He lost all his family. He ended up working as a shepherd during the war and eventually landed in the US. So, and he never published another paper, actually. But this was a breakthrough because it provided an intellectual approach to the problem of emergent properties. So now, if we turn our attention to the brain, and you think about the nervous system, in particular the cerebral cortex, which, as you know, covers the top of our encephalon and is the largest part of the brain in mammals. And wherever you look in the cortex, or anywhere in the brain for that matter, you find neural circuits that are built with gigantic numbers of neurons, astronomical numbers of neurons, and they're all interconnected. And if you look at evolution, you appreciate very clearly that in the evolution, particularly the mammalian lineage, that the numbers of neurons and the size of the brain is increasing dramatically. Now, if you think a little bit like icing, you said, okay, imagine I want to build a system to generate emergent properties. So what do? Well, you build a system with as many components as possible, and then you let them interact as much as possible, because that essentially can maximize the numbers of these stable states in the system. And if you look at the evolution of the nervous system, this is exactly what nature is doing. It's building a nervous system with more and more neurons to the point of view that's just getting ridiculous. We can barely fit through the birth canal of women as humans. And at the same time, it's connecting each neuron with as many neurons as possible. Of course, you cannot connect one neuron to all the other neurons in the brain. We're talking about 86 billion. But in many parts of the nervous system, this is exactly the strategy that seems to be followed, the logic, to maximize the connectivity and maximize the numbers of neurons. So, so it's very likely that when we look at the brain, nature has found is the mother lode of emergent systems, that the game that nature is playing with the nervous system is essentially to build these emergent physical systems in order to compute or to generate behavior. So now we've been at it for 100 years in neuroscience. We still don't have a general theory of how the brain works. We still cannot predict pretty much anything about the brain, let alone understand mental and neurological diseases. And one reason for that has to do with our assumptions as a field from the very beginning. And if you look at the basic assumption that runs through the middle of neuroscience already for more than 100 years, it's something called the Neuron Doctrine. Okay? The Neuron Doctrine was stated by Ramon y Cajal, and he actually got the Nobel Prize for this. He stated the anatomical neuron doctrine that stated that the unit of structure, the nervous system, is the individual neuron. and his contemporary Sherrington argued for the physiological neuron doctrine that said that the unit of function of the nervous system is the individual neuron. And both Cajal and Sherrington were so confident that they called this not a hypothesis or a theory. This was a doctrine. It was religious belief. This is how things work. This is how things are. But it's very interesting that both Cajal and Sherrington were pioneers of methods that studied neurons one at a time. So Cajal actually developed and perfected the Golgi method that stained neurons one by one in all the glory, all the beautiful drawings you can see here. And with that, he elucidated many fundamental aspects of the structure of the nervous system. And Sherrington was the first person to record the activity of individual neurons, of actually individual axons in the neuron. So both of them used methods that were single neuron methods. So no wonder both of them push the idea that, hey, the brain is just a collection of single neurons. So since then, the logic of neuroscience has been very straightforward. Let's just take the system apart, look at brains one neuron at a time, either structurally or functionally, and to understand how the system works, we can actually record the activity of individual neurons, one neuron in one animal performing one behavior, and correlating that with the behavior of the animal, or let's say in a patient with a mental state of the patient or the disease state of the patient. Now, if you think like icing, okay, you say, wait. This is true if the individual neurons are the unit of function, but if this is an emergent system, okay, to record the activity from one neuron and correlate that with the behavior of the animal, it's a little bit like trying to watch a movie on a TV if you look at a single pixel. Okay. Imagine that the brain is a TV screen with 86 billion pixels, and you're going there recording the activity of one neuron and trying to understand what goes on. Well, you're never going to get it. It doesn't matter if you record that pixel forever until infinity. You will never understand what's playing, because the image in a TV, like this kid jumping, is actually an emergent property, and by definition is not present in individual pixels. It's present in the correlation in space and time and color between pixels. And only when you capture that, you can see the image. And only when you see the image, you get the movie. So, my argument is that neuroscientists have been doing the wrong experiment. For 100 years, we've been trying to correlate the activity of individual neurons with the behavior of the animal or the state of a patient, when we're dealing with a system which, according to evolution, it's obviously been made to generate emergent properties. So if this is not the first time that people have made that argument. In fact, it was already Laurent Dedeneau, a disciple of Cajal, who in 1933 argued from looking at connectivity of the cerebral cortex that most of the connectivity was actually feedback. He called these things reverberating chains. So this is an example in the bottom of a neural circuit which summarizes everything you learn. And he said, you have neurons that receive inputs and generate an output, but these outputs branch and go back to neurons that connect back to the first neuron. So they form these loops, these feedback loops, excitatory feedback loops. And he argued that whoever has designed the cortex, the function of the cortex was actually to generate this endogenous activity. He called these reverberating chains, like a chain of neurons that is continuously getting activated. And the reason, and this was not a minor point, because if this is the case, and this is true for the nervous system, or not just humans, but all animals, then you can imagine that nature has invented a way to generate a state of activity which is autonomous, independent of the world. And by doing that, you can actually use this state of activity, these chains, these feedback, as units to represent the physical world. And this is a breakthrough in evolution, because instead of dealing with the physical world, suddenly evolution has created a parallel universe, a formal world of symbols, And these units are now symbols that can be manipulated mentally instead of manipulating the physical world. This is the beginning of intelligence, the beginning of a mental model of the world. So these ideas from Lorente were actually picked up by Hebb. And you know, scientists like to leave their imprint in history. So he essentially scooped Lorente, he changed the name. So instead of chains, he called the same things assemblies. It's the same idea. You have here a loop of neurons that are connected. So the corners of this graph are neurons and the arrows are the connections. And he argued that these neurons would actually assemble themselves into these neuronal assemblies where activity would flow in a loop, in a closed loop. No? So that was exactly like Lorente said. No? But Hebb added something more to this idea of having these endogenous feedback loops. He said that if you have synaptic plasticity between neurons and proposed this rule of Hebb's rule, neurons that fire together would wire together. This is the way he actually wrote it. Then he said it's unavoidable that if you have synaptic plasticity of this sort, you will end up building these loops, these assemblies. Once you build them, you can use them, for example, to implement memory, associative memory. So this proposal of Hebb has had a huge influence in neuroscience and started to focus the attention of people on the possibility that the unit, let's say, of in this case, of memories, of associative memories, are not the neurons themselves, but these loops of neurons, these assemblies of neurons. Similar ideas were proposed by Moshe Abales, who's in Israel, and in this case, he came from completely different angle. He said, as a physiologist, said, okay, if you look at the hardware of the cerebral cortex, and you look at the connectivity between neurons, it doesn't look very good because the connections are weak. The excited connection between neurons are extremely weak. On top of that, they're stochastic, okay, so most of the time they don't work. And then on top of that, they're depressing. So synaptic plasticity, short-term synaptic plasticity, overwhelmingly depressing synaptic plasticity. It's like, I don't know, whoever has built this doesn't want activity to propagate. In fact, this is the perfect system for activity to die out immediately. Except, and that's where he came up with the proposal, if you have a synchronous groups of neurons that are firing together, he called these things sync-fire chains, as you can see here. They will have enough, if they work as a group, they will generate enough activity together that that activity will propagate and turn on another group of neurons, second group, which will then turn on a third group, etc. So he imagined that activity through this cortical circuits filled with depressing weak synapses will propagate with these groups of neurons, these chains, sync-fire chains, that would then light up one after the other. No? He found some evidence in recordings from awake monkeys that this was the case based on the timing of the spikes. He was recording from a few neurons, and he argued that he was tapping into different points of these sync-fire chains. All of these ideas were given a mathematical backbone through the work of John Hopfield. Now, Hopfield is a physicist that knew very well the IST model. And I said, wait. This problem of feedback neural circuits is isomorphic mathematically with the IST model of magnetism. So he said, if you have a neural circuit like this one, these are the little neurons connected through these axons to these dendrites, and this connectivity is recurrent fed back. In the spatial case in which this connectivity is complete, in other words, that every neuron is connected with every other neuron, and if the connectivity is symmetric, the matrix of connectivity is symmetric, in other words, that the connections from neuron A to B are the same as the neurons from neurons B to A, it proved that this was exactly mathematically the IST model. And he casted this equation on which he defined the term energy, in which this equation is just like the IST equation. And here you have these V's represent at the activity of the pre and postsynaptic neuron, and the T, the coupling coefficient, now represent the synaptic strength between these neurons. And if you sum across all the activity of all the neurons and all the synaptic strength, and you define this negative term of energy, which again defines the propensity of a system to be stable. So the more connected, the stronger the connections. If the firing is synchronous, then the system is going to actually be very stable. because they have very little energy. And he imagined that it would settle in these states that he called attractors. Now, this is a 3D representation of a multidimensional map in which every dimension represents the activity of one neuron. But you can visualize these attractors in 3D. As poor humans, we cannot see more than 3D. So imagine that every point in this map represents the activity of a network at a particular point. So for instance, imagine that this corner is with all the neurons are on, and this corner where all the neurons are off, and these are some intermediate states. Now, this topo map, the height represents the energy of the system. So these lower valleys are what he called the attractors, which are the states of the lowest energy, according to this equation. You can compute that energy. And he argued that these attractors were going to be stable state or semi-stable state in the life of this neural network, and they could be used to implement memories or to implement solutions to computation. In fact, he argued that if you imagine a ball rolling down the landscape, so that's actually your brain at any particular state, and as you think, the ball is rolling until it falls into an attractor. And that attractor could be a memory, the memory of your grandmother, or it could be a computation if you're trying to compute 2 + 2, and this attractor is the number 4. So he argued that the dynamics, the ball rolling through the landscape, is the computation itself, and the attractor is the solution to the computation. And because of this energy structure, which is intrinsic to the system, this is not something that we impose from the outside. This is something that the system has because it's built with this matrix of synaptic connections. So these states had pattern completion, which means that if you want to get to the attractor, you don't have to push yourself all the way. You have to just get close to the lip, to the beginning of the valley, and then the system itself will bring you down, and it will complete the pattern. So these are some of the ideas that have been proposed in terms of what could be the emergent states of neural circuits, but they all agree on the same thing, that you could have these synchronous groups of neurons, whatever you call them, whether you call them chains or assemblies or synfires or attractors, it doesn't matter, the nomenclature. And then this could be used to compute, they could be used to generate a memory, to generate maybe a behavior, to trigger a perception. So how can you now test all these theories? Well, just going back to icing and to the TV problem. Well, so it's very simple. This is the cerebral cortex of a mouse, the primary visual cortex. This is a coronal section. Every one of these dots is a neuron. So what you have to do, you have to record from every neuron. And you cannot do it with electrode. You're going to turn this thing into Swiss cheese. So, what we did when I was a graduate student with Larry Katz, and this is the Whistle lab back in the late 80s, we explored the possibility of performing these measurements optically. And we focus on calcium indicators that have been developed back then by Roger Chan, which are molecular dyes that bind calcium and change spectroscopic properties when they do that. And we found by chance that you can use these calcium indicators, a particular form of them as etoxymethyl esters, and label brain slices with these calcium dyes. In fact, this is a brain slice that has been labeled with calcium indicators, a living brain slice. So you can see that this labeling is very complete. In a good day, you can label 95% of the neurons using these calcium indicators. And then we had a stroke of luck. Well, the first piece of luck was to see that you can actually label all these neurons with calcium indicators. But the second piece of luck is that if you measure the fluorescent intensity as a function of time, you could see these neurons blinking on and off. And I remember when I first saw this, I told Larry, look at these movies of calcium in the neurons. The neurons are blinking. And he said, Rafa, the arc lamp of the microscope, it's actually this microscope here, has been changed, has not been changed in a long time. So why don't you go and change it, because the arc lamp is flickering. And then I told Larry, the next experiment I did is I put TTX, the sodium channel blocker, and that blocked all the blinking. And I said, Larry, this is very interesting. The arc lamp is sensitive to TTX. Anyway, so he laughed and he said, and you have a thesis. And this is how we discover that you could use measurements of intracellular free calcium with these calcium dyes to monitor action potential activity, because every time the neuron fires an action potential, as you can see here in these electrical recordings, there's an increase in intracellular free calcium concentration because the calcium channels in the membrane open. And the calcium concentration is so low at rest that you can, with high affinity calcium indicators, you can actually measure this calcium influx that comes because of a spike. So that meant that we could start making movies like this one. This is 500 neurons in a brain slice, primary visual cortex, the mouse, spontaneous activity. And here we've colored the neurons that are undergoing these spontaneous increases in calcium concentration with red. So it was very exciting because we could start to see the activity of groups of neurons together. Now, the mouse primary visual cortex has about 180,000 neurons, and the mouse brain has about 100 million neurons. And here we're only looking at about 500 neurons. Okay, so you can say, well, you're still not looking at the TV screen, Yuste. I said, that's true, but in this little corner of the TV screen, these 500 neurons, we can see every neuron. So that enabled us to start exploring the presence of these groups of neurons, these attractors, these neuronal coactivations for the first time with the imaging method, with light. And to do this well, we also needed to be able to look deeper into the brain tissue. So these experiments I showed you earlier were from the cut end of a brain slice. But I was lucky I worked with Winfried Denk when I was a postdoc at Bell Labs. And Winfried had invented this two-photon microscope. I'm not going to have much time to discuss it, but it uses infrared light, and it actually generates very high concentrations of photons that can trigger a double hit, where a molecule can actually get excited by two incoming photons of infrared light and generate an emitted photon of visible light, fluorescent photon. And with these two-photon lasers, there's two advantages. One, that infrared penetrates deeper into brain tissue. And the second advantage, that this is a reaction which is nonlinear. The rate here depends on the square of the concentration of photons, because the factor of 2 over here, which means it only occurs at the focal point. So with Denk, we demonstrated for the first time that you can actually use calcium imaging to image neuronal activity, actually activity of even these parts of neurons, dendritic spines, in the middle of brain tissue. So with calcium imaging and with two-photon, we then started looking for these emergent properties of cortical circuits. And the first thing we found back in 2003, I was with Rosa Cosar, who was a postdoc in the lab, and looking at brain slices, she found with two-photon excitation, she found this coherent activation of groups of neurons that you can call them ensembles. The nomenclature, it's all over the place in the literature. It doesn't matter. We're all talking about the same thing. And ensemble is a group of neurons that are firing together. Let me show you an example. So these are now, this is the calcium raster plot. Every line is a neuron as a function of time. Okay, and the little black dots represent whenever that neuron is active. So this is all the activity in 750 neurons over a period of 10 minutes in a brain slice. And you can see that there's groups of neurons here that are firing together. Do you see these little lines that run through it? So these are the ensembles. There's a group of neurons that is synchronized. Suddenly, for reasons we don't understand, they fire together and then they essentially, activity goes somewhere else. This is exactly what Hopfield had in mind. The ball rolling into an attractor and then moving on to another attractor. You can start to quantify these ensembles if you collapse this raster plot into a histogram, the percentage of cells that are active as a function of time, and you can see this is very few numbers of cells, like 7% in this case, are active together. And you can just set up some sort of threshold to distinguish it from noise, from background activation, and this is what we call an ensemble. And these ensembles had very particular properties. Some of them were very localized, like this case, to a particular layer. Look at this one here. It's exquisite. All these neurons in red are the ones that participate in one ensemble. But most of them Some of them were columnar, but most of them were dispersed throughout the tissue. Something very interesting happened, and that's we started making these movies in awake behaving mice. And here you have again, sorry, just see if I can play the movie. Here we go. So here you have two-photon calcium imaging of groups of neurons in primary visual cortex of a mouse that's actually awake behaving, looking at a screen. And if you look, this is the raw data. You can see that there are groups of neurons that are firing together. This is the analyzed data from this movie. And you can see, if you look at this movie carefully, that these neurons are not firing individually, they're firing in groups. You see how they go together in little earthquakes? This is exactly what we're talking about, these ensembles. So this was not a slice artifact. This happened in awake behaving animals. And if you can see, these ensembles are occupying similar cortical territories. They're in the middle of each other. So you can have neurons that sometimes participate in two ensembles, and you have ensembles that are located on top of each other and involve slightly different neurons. So how do we analyze this data? So with a computer program, we identify the position of every neuron. And then for every neuron, we measure the fluorescent intensity as a function of time. Again, this is all calcium imaging, two-photon calcium imaging. And we, using an algorithm, we calculate this probability that the neuron has higher action potential during this frame. And we build a raster plot. Again, every line is a neuron, and every one of these black dots is whenever the neuron had at least statistically defined with very conservative statistics, an action potential during this period. And you can see, just like we saw in slices, that the activity is actually going down in these little stripes, which if you plot it here as a histogram, you can see these little earthquakes, and these are the ensembles. So in our hands, in our lab in New York, the activity of the cortex in awake behaving animals is dominated constantly by these groups of neurons that are ensembles that are firing together. And you can see here an example of one of these ensembles. You just pick one of them, and this is how it looks like. And if you pick another one, you will see another group of neurons in the same cortical territories that are firing together synchronously. So, so when we show the animal a visual stimulus like high contrast gratings, like here, you can see in red the neurons that are participating in these ensembles, and you can see that most of the activity that's present in the cortex is in the form of these ensembles. And the same thing happens if the animal is looking at a natural scene. In other words, a movie, not of gratings, but for instance, a BBC documentary. This is normally what we use of nature to try to approximate the statistics of natural scenes. And when we analyze the data, again, with very conservative statistics, the majority of the spikes that these neurons are putting out are actually always parts of these ensembles. They're synchronized in these little stripes of action potential that run down the raster plot. So this was very exciting, but now look at the top. This is now spontaneous activity. So this is either in the dark, okay, there's no visual stimulus, or with a visual stimulus like a gray screen, which doesn't change. And even in those circumstances, we're imaging the primary visual cortex, okay? Even in those circumstances, the majority of the spikes are participating in these ensembles. So that means that the cortex is generating continuously these ensembles of neurons, whether it's spontaneous activity or whether the cortex is actually engaged in the visual stimulation. Moreover, if you analyze the position of these ensembles that you see spontaneously, so these are examples of spontaneous ensembles. This group of neurons is one. This is another one, another one, another one. This happens over a period of maybe 10 minutes. And then you analyze the ensembles that happen with visual stimulation. These are now ensembles that happen with visual stimulation. It turns out they overlap. They're the same statistically identical groups of neurons. So you can see, for instance, these neurons that have a little green circle are the same neurons that are appearing here together in this ensemble. And this ensemble here is also reappearing in this frame over here. Okay, so So two things. One, ensembles never repeat exactly. Whenever you catch the same ensemble, you see a group of neurons that are repeating and another groups of neurons that come and go. There's always a variation, as if the activity of the cortex is never completely the same. There's always some change that has happened. But the second thing is that the evoked ensembles activate endogenous ensembles that are there already. So this led us to hypothesize that the building blocks of cortical activity are these ensembles, these coactive ensembles. And then when you have visual stimulation in primary visual cortex, you activate, you build this response using this intrinsic lexicon, like these words in a dictionary that are there already. You essentially line them up in particular ways depending on the stimulus that the animal is seeing. So this was very exciting, but this just only showed a correlation between, let's say, visual stimulation and these cortical ensembles. That doesn't mean that these ensembles are actually functional. In order to test if these are functional, you need to actually demonstrate causality. And for us, this means what we call the piano experiment. So what do we mean? Imagine that the cortex is like a piano. Every neuron is a piano key. And then you can go in and turn them on at will in any spatial temporal pattern that you want. So this is what we need to do to the cortex. So the dream experiment is to image ensembles. This is an example, one ensemble, another one, as the animal is doing some behavior. And then we turn on our optical piano and we activate these neurons in the same order with the same precision that nature build these ensembles. And then we can ask the question, what happens to the behavior of the animal? Is there a causal relation between us playing the piano, playing these ensembles in the cortex of the animal and whatever happens to the behavior of the animal or to the cortex or to the activity in the cortex? Okay, so to do this piano experiment, we need to do two things. We need to measure and we need to activate. But one problem with these ensembles is that I showed you earlier that they're on top of each other. Okay, so you can have one ensemble like these fingers in my hand, and it's activating neurons, and there's another ensemble in between. Different neurons, some of them are shared, so it's actually even worse. So you have to have single-cell precision to turn on some neurons, but not their neighbors, which means you have to do all of this with two-photon. The activation has to be done with two-photon, but it has to be done with a way in which you can actually, at will, just like my fingers when I play the piano, turn on some keys and not others. So our piano is something that we call spatial light modulator. This is a holographic device. It's a liquid crystal, an SLM, in which we can actually build any arbitrary pattern of light. So this is an example of a word that we want to write with light, with two-photon light. This is the face mask. This is essentially the Fourier transform of this word that we upload into the SLM, and this is what comes out the other end. So this is actually the two-photon words written with this holographic piano. So the student who developed this holography two-photon microscopy in my lab, Nikolenko, had nothing better to do than paint pictures of famous neuroscientists like Rafa with two-photon light and project them back into the cortex as if the cortex would care. Anyways, this is what happens to the graduate students when you're not looking. So then we had it all. Then we had two-photon calcium imaging to be able to measure the activity of the groups of neurons, these neural circuits. And then we have this holographic piano to turn the neurons on and off. But in order to do that, we use an opsin, a particular opsin called C1V1, developed by Dijsselhoff. And this is a channel opsin that is very slow, which means that it generates a big current, which means that it can make neurons fire with very little bits of light, which is what you generate with two-photon excitation. And this is an example in which we can actually are playing the piano with two fingers. We have two neurons near each other, and using this holographic two-photon optogenetics, we can turn one neuron or the other neuron or both of them together with exquisite single-cell precision. These neurons are on top of each other, 20 microns away. This is the worst possible case, neurons on top of each other, and we can turn the top neuron or the bottom neuron without any contamination. So we started doing these piano experiments with two-photon holographic optogenetics in primary visual cortex. And for that, we needed a setup with two lasers. One is the two-photon calcium imaging laser, and the other one is the stimulation laser that goes through this spatial light modulator that generates the pattern that we want to, this is our piano, the one we want to turn on in the neurons. And we're doing these experiments through the skull, the thin skull of the animal, in an awake behaving animal, while the animal is viewing a visual screen. And the first experiment, this was done by Luis Carrillo-Reed, who was a postdoc in the lab, was to play the piano with our elbow. These are very hard experiments. A lot of things have to work. Okay, so so we were saying, okay, let's try to turn all the neurons in the lower part of the visual, in this left side of the visual field. And this is an example of two-photon calcium imaging, and Luis is optogenetically activating all these neurons here, as if, again, if you're pressing the piano with all the elbows. And we were pretty happy because through the thin skull, you can actually see the calcium activity of these neurons when we turn them all on. And then came the discovery. So Luis comes to me one day and said, Rafa, do you remember these neurons that were that I was making them fire together by playing the piano with my elbow, sort of doing optogenetics in the lower part, in the left part of the visual field? I said, sure, yeah. I said, well, guess what? They're still firing together now on their own. I said, what do you mean? Yeah, so it looks like without knowing, the minute that Luis pressed down on these neurons 50 to 100 times together, they stuck together and they started firing spontaneously, becoming an ensemble. So he imprinted these ensembles with optogenetics. So this, moreover, these imprinted ensembles continue to fire together even a day later. So 50 to 100 times activation can rewire this neural circuit in a way that you build an ensemble, an artificial ensemble, you imprint it, and that group of neurons starts to fire together. You can see it here. So this is the imprinting, and this is the ensemble that we built up, and it's firing spontaneously here and here. And then overnight, you look at this piece of cortex again, and these neurons are still firing together. So that means that we actually generated, without us knowing, an artificial ensemble. But when Luis started playing the piano with a finger, he found something even more interesting. So now he's using the SLM to turn the neurons on one by one. In a way, we're sort of playing. We're literally playing the piano, just having fun there. And when he activates this particular neuron, the neuron turns on. So he was very excited. Again, he's doing this through the skull of the animal with this holographic optogenetics and turning the neurons on one by one. But he found that if this neuron that he was turning on one by one belonged to this imprinted ensemble, about 30% of the neurons, when he hit them and turned them on, triggered the entire ensemble. And this is illustrated here. So here you have a raster plot, about 55 cells as a function of time. And in these pink lines, he's turning on one particular neuron, which is neuron 25, that belongs to this ensemble of neurons that is in pink. Okay, and this is neuron 25. And often when he turns out this neuron, nothing happens. Like here, nothing happens. But look at this case. He turning on this neuron, it trips all these other neurons, which happen to be the neurons that belong to this imprinted ensemble. So this demonstrated in front of our eyes pattern completion, making Hop feel very happy. You can see these ensembles, these attractors, you can build them, and then they have pattern completion. You trigger one and you trip the entire thing. No? You complete the entire ensemble. Now, these were all artificial ensembles. And pattern completion was, again, the idea is, as many theories have proposed, is that you can activate a group of neurons together, they somehow reinforce their connectivity, and then you can trip the entire thing. This is exactly the Hebbian assemblies hypothesis, the Hopfield attractors. You can activate one and trip the entire group. Okay, so now this pattern completion was still present a day later. Okay, so this is not a little phenomenon. This is super robust. No, you build these ensembles with 50 stimulus, and then one or few of these neurons have pattern completion property. You turn them on, and you can do that here, turning on this particular neuron 19, and you treat the ensemble. This is the analysis of the ensemble. I won't get into that. And these are the analysis of the single cells. And you can see this ensemble that is lighting up on day one. And if you turn on this neuron the next day, you can still treat the same ensemble. So pattern completion is there even a day later. This is permanent, or at least 24-hour long plasticity that we've induced. So very exciting, but these are all artificial ensembles. We're putting them in. Okay, so how can we test if these ensembles are for real, if they actually matter for cortical function? So in this last series of experiments, we did behavior with the animal. So the animal was performing a behavior. This is the piano, the dream piano experiment. We measure these ensembles, and then we play them back, and then we see what happens to the behavior. And we can test the question if ensembles are functional units. And this was done with an animal that's trained to lick when he sees these vertical bars of light. You see how the animal is licking. It's a go, no-go task. And then we show the animal horizontal bars of light, moving gratings, and we train the animal not to lick. You see how the animal is not licking. They learn these tasks very quickly. In a week to 2 weeks, they're up to 90% performance. And simultaneously, we're doing two-photon calcium imaging to measure the ensembles that correspond with one stimulus or with the other stimulus. The experiment is very straightforward. Again, visual stimulus, vertical bar, go, licking, and we measure these ensembles, let's say these blue cells. Horizontal bar, no go, no licking, and we measure these other ensembles that correspond to the horizontal stimulus. And then we turn off the visual stimulus, we turn on our SLM, our piano, and then we play back these ensembles and see what happens to the behavior. So let me show you what happens. The first experiment we did was to activate the no-go ensemble when we were showing the animal a go stimulus. So this is a behavioral measurement. Licking is in blue and the stimulus are in red bars. So the short are the no-go and the long, sorry, the short are the go stimulus, and every time, not every time, but most of the time we show him a go stimulus, the animal licks. And the long bars are the no-go stimulus. It's pretty good. Here he doesn't lick for whatever reason, and then he goes back to licking. And then, sorry, I got this wrong. So we're showing him the, yeah, no, I got it right. And then in the middle of this, we turn on with our piano the no-go ensembles here. And what we find is that the animal stops licking. Now think about that. We're turning on about 10 to 20 neurons in the middle of the brain of this animal that has 100 million neurons. And we can stop his licking behavior dead. No? Okay. This is illustrated here. This is the raster plots. Actually, probably you should look at this panel E, the performance of the animal when the animal is trained. It's pretty high performance, from 70 to close to 100% of the time he licks to the go stimulus. And when we turn on the no-go ensembles, you can see how the performance statistically degrades. No? So we're modulating the licking by turning on the no-go ensembles. Okay, the next experiment was the opposite. Now we show him a go visual stimulus, but we actually lower the contrast so the animal barely sees that. So this is one advantage of working with a visual system, that you can actually control the stimulus very precisely. And with low contrast, we make it very hard for the animal to see the stimulus, and the animal doesn't lick. So here we show him low contrast stimulus, the animal doesn't lick. In the bottom, we're showing him this low contrast stimulus, but now we're activating the go ensemble. And look what happened. Now he sees He sees the go stimulus. Now he licks every time he sees this low contrast go stimulus, which he wasn't seeing before. And this is illustrated here. The performance of the animal to low contrast go stimulus is very low. Okay, and then when we actually activate with this holographic piano, the go ensemble, the animal licks as if the stimulus was high contrast. Now the final experiment is told you, we turn off the stimulus altogether. We only now play the piano. So no more visual stimulus. And now we play the go ensemble in the absence of any visual stimulus. And to do that, we actually activated pattern completion neurons. So instead of turning all of the neurons, we just went for the ones that were pattern completion. And we couldn't do it with one, but when we activated two pattern completion neurons, illustrated here, this neuron, for instance, 9 and 8, this is a raster plot of all the spikes in a group of neurons. When we turn them on, most of the time, nothing happens. But here, when we turn these two pattern completion neurons and we trip the ensemble, the animal goes and licks. Okay, so the animal is licking because we turn on an ensemble that corresponds to the go ensemble, and we did that by turning on the pattern completion neurons. And you can see in this graph the performance of the animal when we're recalling the go ensemble is up to 70%, 80%. No? As when we're not recalling the go ensemble, the animal is not licking. And this is the distribution on spatial distribution of the go ensemble with the pattern completion neurons that we're activating. So this means that turning on the go ensemble triggers the behavior. This is causal link. No? And then the final thing is something what we observed in the spontaneous activity of this animal between trials. We notice that by chance, if you look at this panel on the right, the neurons that were part of the go ensemble became active spontaneously. We were not playing the piano. He was not looking at a visual scene. They just went like that, they popped. And when that happens, we only see that in one animal, but it's very interesting. When that happened, the 3 times this happened, the animal licked. So the blue bars is when the animal licking. So this is now spontaneous activation of the go ensemble, intrinsic activation of the go ensemble triggers licking. So I'll finish with the conclusions. I've shown you evidence for emergent properties of cortical circuits using these methods, two-photon calcium imaging, two-photon holographic optogenetics. We see that cortical activity is organized in groups of neurons firing together and ensembles. You can call them attractors. These are spontaneous states. Neurons can participate. The same neuron can participate in different ensembles. I didn't show you this. About 20% of the neurons are promiscuous. The dynamics, the activation of these ensembles is repeated, can repeat, but it's never exactly the same. There's like a core of neurons and then non-core groups of neurons. These ensembles show pattern completion. The visually evoked ensembles are statistically indistinguishable from the spontaneous ones. We can imprint artificially, playing the piano with our elbow, these ensembles, and we can recall them for several days. This is what a physicist would call a phase transition. This last part of experiments show that ensembles are sufficient and necessary for behavior. If we turn off the go ensemble, behavior stops. If we turn it on, the behavior happens. And again, this is, it was hard for me to believe this experiment from Luis because he was turning on 2 pattern completion neurons, 2 in the brain of 100 million neurons and generating triggering behavior of the animal. So this is quite extraordinary, I think. Our hypothesis is that these ensembles are actually functional units. In fact, we would argue that these are internal units of perception. And here I distinguish between sensation, which is the response of the neurons in your brain to a sensory input, and perception, which you can describe as your internal interpretation, your thinking of that external input. This is something that has been discussed in psychology and philosophy. In fact, Kant thought of these things already, and he argued that that perception was based in what he called representation, Darstellungen in German, of the stimulus, and we think these ensembles could be the Kantian Darstellungen. And this also shows a very close link between the methods and the paradigms. Because only when we use these methods to look at the entire activity of the entire cortex, you can then see these emergent properties. Wanted to highlight the work of the lab. I talked to you about the people that did the experiment. The last batch was done by Jim Miller and Luis Carrillo-Reed, both postdocs. We're funded by US federal funding agencies, and I have a conflict of interest. Columbia, through our lab, has the patent for this piano two-photon imaging and photostimulation, holographic two-photon imaging and photostimulation. And I just wanted to finish with a quote from Sidney Brenner, that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. Thank you. I'm happy to take questions now.
Thank you very much, Rafael, for your nice presentation. Since we are doing a webinar, I have some question in the chat box, but we can also ask the people directly to ask them. If somebody would like to raise their hand, just do it now, and I can unmute you, and you can ask the question directly to Rafael. So Todd, I'm allowing you to talk now, and you have to unmute yourself, and you can ask a question. Allow to talk. Okay.
Okay, can you hear me?
Yeah, perfect.
Yes, perfectly.
Thank you. Great, wonderful talk. Just had a question. So it is remarkable that you can manipulate, such a small set of neurons compared to the number of neurons in the cortex even.
Yeah.
That this manipulation can produce such a clear behavioral phenotype.
Yeah. Isn't that incredible? I mean, I had a hard time believing this data when Luis showed it to me. I said, no, this is impossible. You cannot do that. No.
So this, to me, has major implications on redundancy in the brain. That things are not very redundant. If you can take out with this little window that you're looking at, you can sort of, have such a large effect. And so I'm just curious your thoughts on.
Well, I think Alex got it right. So I think this is just this synfar chain he was talking about. In fact, with our experiments and with pattern completion, we can now explain how this happens. So we're turning on this first ensemble, the Go ensemble, and by turning on these pattern completion cells. Imagine that these neurons in the Go ensemble are connected to a whole bunch of other ensembles throughout the brain. And if they happen to hit a pattern completion neuron of the second ensemble, they can trip this second ensemble. And then again, these red cells here would be pattern completion neurons. So this is actually what we add to Abeles's theory. In fact, we demonstrate this directly experimentally. So I imagine that turning on these two neurons in the primary visual cortex sets up like a series of avalanches of synfar chains that propagate through the brain, and eventually there's some that control the licking of the tongue. And that's how I can intellectually explain how the activity of two neurons can have such a huge effect in behavior in a complicated brain.
Great, thank you.
Okay, thank you very much. Shinjin, allowing you to talk.
Actually, let me share with the audience this announcement of an optogenetics meeting that we're going to have online in a couple of weeks, May 27. It's actually free. It's a whole day of talks, and many of the talks have to do with this piano type of experiments. We're going to have great people, Peter Hagemann and Karl Deiserhoff, the pioneers of optogenetics, with a whole bunch of speakers from Europe, the US, and even Japan. Yep.
So we have a few more questions.
Can you hear me?
Yeah. Yes, perfectly.
Okay. Hi, I'm Shinjini. I'm a postdoc from the University of Edinburgh. And I was interested in the phase transition experiment that you did where you activated this entire group of cells. I was wondering if it's a thought experiment more than anything else, but does that alter the learning of the animal? And does it have an actual effect on the animal's ability to learn things?
Yeah, we haven't looked at the ability of learning, but the generation of an artificial ensemble doesn't seem to change the existing ensembles. So it's like you push one. If you think of this as a Hopfield landscape of attractors, you're just throwing in another one in the hole, but there's many of them. But my prediction is that it's going to depend on how many of these additional attractors you build in. At some point, you're going to start affecting the existing ones. And in that case, you could actually alter learning, I think, because you will distract the existing landscape, so to speak. In a way, it's like destroying the mental picture that the animal has of the visual world.
Thank you very much. Thank you for the talk as well. It was incredible.
Thank you.
We have three more questions that Peter, Jonas. Yeah.
Actually, I see many questions about technical details of the experiments. These last experiments were published last year in the journal Cell, exactly a year ago. If you just look us up in the even in our webpage, we have the PDFs of all these papers freely available. You can look up all the details of the experiments.
And one more comment, this talk is being recorded and we will put the link online. And so you can, if you're interested, look it again.
So now, Rafa, very nice talk. So if a single neuron is sufficient to recruit an ensemble, this means that the output synapses of that neuron are detonator synapses, or how do you interpret these findings at the synaptic level?
Yeah, we're actually looking at this right now. We're trying to understand the mechanism of pattern completion and the mechanism of ensemble formation. We're not completely finished with our experiments, but the bottom line is that, believe it or not, it's not Hebbian, okay? So when we build these ensembles artificially, we were sure they were Hebbian because, hey, this is head. You turn the neurons together and they stick together in an assembly. And this is just like we demonstrate. In fact, we finished our science paper in 2016 saying that with the sentence that this demonstrates the Hebbian principle. We may have been wrong. We think that this is due to hyperexcitability, that neurons that are stimulated together, they become hyperexcitable, so they're closer to threshold. So doesn't mean that the synapses are stronger, but we think that the effect of the same synapses is weaker, stronger, as if it's pushing an iceberg out of the water. So this is what we call the Iceberg Model. In fact, you know what? I can even share with you the figure that we're making for this paper. Let me see if I can actually find the figure. This is one advantage of giving talks online. Here we go. This is the Iceberg Model. Here we go. So imagine that this is the connectivity of a piece of cortex before and after we do this imprinting. And after the imprinting, and the neurons in red are the ones that are firing action potentials, and the neurons in gray are subthreshold. So we don't change the connectivity, we just make everyone more trigger happy. So it's like we're pushing this iceberg out. And then this neuron would suddenly trip a whole bunch of neurons that are very close to threshold. So that's sort of our current model.
Thank you.
Do we have time for two more short questions? Artem and Venkat, allow you to talk. Artem?
Hello, can you hear me?
Yes, perfect.
Yeah. Thanks for the talk. I have, I think that was the technical question that you said that we can check on the paper, but so how can you treat the recording that been kind of perturbed by this stimulation that you do? So if you have the optical path that combines both lasers, how can you then differ between this signal that you get?
So I'm not sure if I understand you correctly. So we're imaging with one laser and stimulating with another laser. They have different, let me just share my screen here. Actually, I have to tell you that there's some advantages for talking online. I have all my slides here from all the papers, everything here. So yeah, so this is technically how we do it. So there's no crosstalk between both laser lines. One is at 940, the other one 1064. So that means that we don't activate the opsin with the calcium imaging laser, and that's critical. And then we can then, as I show you here, you can actually, let me show you here. It's an example here, maybe. This is maybe a better example. Here you can see very clearly. No? You can see the resting calcium level in all these neurons, and you can see when we're actually turning on these other ones with the second laser. And you don't see the second laser, you just see these neurons lighting up. So that means that it's very, very little, or here we go, no discernible crosstalk between both laser lines. We do this with one SLM in these experiments. We're using one SLM, but we can put a second one here, and then we can do the thing in 3D because this hologram has 3 dimensions. In fact, if we look at our papers in PubMed, some of the last papers that we publish are actually doing 3D optogenetics. So we do this piano in 3 dimensions, and we can image in 3 dimensions. We can, of course, these ensembles, I show you only 2D data, but these ensembles, they have 3 dimensions. So if you really want to get into the game and play the piano, you have to play in 3 dimensions. You have to do it the same way that nature does it.
Okay. We have a last question. Venkat?
Yeah. Hi, Rafa. Greetings from India. Great
Hello,
talk.
hi.
And I was wondering.
Yeah, I love the ability to connect with all over the world. This is the best thing we could do to put ourselves together in the middle of the pandemic, not to show that science goes on, virus or no virus, and we do that better than, more connected than ever.
Yeah, indeed. I was wondering if you think that these sorts of cell assembly mechanisms underlie all of cognition, or do you think they are restricted to things like learned behaviors or visually evoked tasks, etc.? Yeah.
I think these ideas, which are not mine, again, I credit Lorente, I credit Hebb, Abeles, and Hopfield, no? And other people, many other people that have said very similar things. I think this is a general theory of how the brain works. I would go back to the neuron doctrine and change it. In fact, I've argued for it wrote a review pushing this idea that we have to let go of the neuron doctrine and start thinking of ensembles and groups of neurons and thinking of these circuits like neural networks, recurrently connected neural networks. So I think it's a very general theory, and it actually explains, when you have a new theory, you have to explain everything that the old theory explained and the things that the old theory couldn't explain. So this new theory can explain all the single neuron data, because there are special cases of these parent completion neurons triggering ensembles. But again, this is a hypothesis, it's not a doctrine, and we're in the middle of trying to test if this hypothesis is true, not just in visual cortex, we're also doing experiments in prefrontal, in hippocampus, and I know colleagues from all over the world are testing these ideas in other parts of the brain as well, in mammals and other animals.
Thank you.
So thanks a lot, Rafael, for your nice talk again. We were almost above 300 participants all over the world. So it is first time I'm doing this as a host, and I'm really glad that it worked out well.
Good job.
Thanks to everybody for tuning in, and have a great, what day is today with quarantine? I don't remember. Tuesday.
Yeah, Tuesday.
So, Rafael, you're going to go to online meetings with the students, and I'm going to finish up in the meeting right now. Thanks to everybody.
I switched to the other meeting. I don't stay in this meeting, no? Correct. Okay. Thank you. Thanks for all your help.
Thank you very much.
Cheers. Stay safe.